Yes, I started.
Perfect. Yes, I'm ready to start. If everyone more or less are here.
Yes, go ahead.
Okay. Yeah, perfect.
Perfect.
Thanks everyone for being here to the Computational Modeling and Digital Twin Technology in Biomedical Engineering webinar. This webinar is linked to the special issue that for which I've been, I was guest editor. And before starting, a big thank you to the editor-in-chief, Dr. Paolo Bonato, and his teams, in particular with Cara MacArthur, Cara MacArthur that is the managing editor, and all the team, Federico and Virginia, right? So just a few words about myself. I'm Mayana Laola. I'm assistant professor at University of Nottingham. I work at the School of Medicine in anesthesia and critical care research. I use and develop a high fidelity, highly integrated computational model of pulmonary cardiovascular systems with the principal aim to configure in silico and therefore virtual patients who are normally admitted to the intensive care units. I develop strategies, tailored and optimized strategies in terms of ventilation and cardiopulmonary resuscitation of these patients. Before we start with the talks of the webinar, just a brief overview on the special issues. All the papers we're going to publish, they are already published, some of them, the majority of them are already published, and the special issues include 7 papers that all of them use computational modeling, but for different application, or better, for application in different areas of medicine, as critical care, psychiatry, psychology, cardiorespiratory, cardiovascular system, and otolaryngology. The upper panel that you can see here included the 3 papers, the 3 studies that will be presented soon after in a short time. Here we go. We have Dr. I'm so sorry for the mispronunciation of your last name, Constantin, Sharudolev. I'm sure that I made mistake, but the authors of this paper, they come from Germany and United Kingdom, worked on mechanistic virtual patient modeling to be used to capture feature of patient status and dynamics while reducing biases introduced by noises intensive care datasets. The second talk in this study are from a group that are working at Meet Lincoln and at Harvard, and the authors here, they will be Professor Quatieri and Dr. Wang. They used and developed a digital emotional twin that can be used to select the most salient temporal region of a speech to improve post-traumatic stress disorder detection. The third talk is from a group that works at New York University and at Houston University. We have here today Samon Kazemi and Professor Rose Faik. I'm sure I made a mistake as well in this pronunciation. In this study, the authors use Bayesian filtering approach to track the hidden status during the handbag task during calm and exciting music. We will see all details in a few seconds. This study, instead of on the cardiorespiratory, in the cardiorespiratory system, is presented by a group based in Lahore, Pakistan, and Western Washington University. It's a patient-independent approach for the estimation, the continuous estimation of respiratory rate and blood pressure, using a spectrotemporal feature and using one-channel photoplethysmography. So they are using a very non-invasive methodology. Regarding the cardiovascular systems, this study is presented by a very cross-institutional group based in Kingdom, Saudi Arabia, Southampton in the UK, and Paris in France. Here the authors have used, have apply the fractional order calculus in modeling to model arterial compliance in human vascular aging. The last published work is from a group all based in the UK at Sheffield University, and here the authors use the computational model to simulate the impedance spectra of thyroid and parathyroid system and how they are influenced by difference in composition and morphology. The last paper, just accepted yesterday, is a beautiful review on digital twins of the biological system. The group is based in the United Arab Emirates and explored the potential application of digital twins, going from definition, history, and the benefit, risk, and challenges of this application, of digital twin application. So thanks a lot for being here. Just this is the brief overview, and now we are ready with the talks. I will stop share my screen. Perfect. If the first author, the first colleague that can share the screen, Constantin, we are ready with your talk. Please, you can make your question in the chat or after the talk.
Just a second, because I had
sure.
I had to just allow.
No worries.
Yeah, just a second. I had to allow to share my screen in Zoom, so I probably have to leave and reconnect in a few seconds, if it's okay. Just a second.
Yeah.
Okay, so hopefully now it works. Okay, can you see my screen now?
Yes, I can
Thanks
see
a
it,
lot,
yeah.
and sorry again. So yeah, it's then the double pleasure to kick off today's webinar for me as well. So just very thanks for Professor Laola, for Professor Bonato for inviting me and giving me a pleasure to introduce my work that I done during my PhD studies at the University of Aachen. So now I'm already acquired my doctor degree, and now I'm working in the Bayer and in the informed drug development department. And actually our group has been recently also renamed in the virtual patients. And in this group we have a small subgroup who are working with the real world data. And the paper I'll be presenting today is a computational simulation of virtual patients that reduces the dataset bias and improved machine learning based detection of IRDS from heterogeneous ICU datasets. And just to give you, because there's so many different notions in the title, it's quite complicated. So let's just start with just explaining this one by one. So what's intensive care unit setting for those who are familiar with it? It's quite really data heavy and then data intensive domain of medicine, of care. And exactly because the patients that are admitted are monitored quite heavily with different devices, and for instance lab measurements are being taken, and interventions, medications, vital signs, everything is recorded, and therefore it's quite a nice domain for the application theoretically of different data-based tools like machine learning, for instance. And there also an urgent need for these approaches, for instance, to predict, as one of the examples, to predict, to forecast critical states like septic shock, or acute respiratory distress syndrome in this case, which actually develop over hours, on the hour or daily scale. So these are very critical, very morbid states that actually have a high morbidity and mortality, and therefore is an urgent need to develop an early warning systems and predictive models for this. Actually, yeah, there's this, and also it's quite. So this domain is quite nicely fit for the application of data-based models. However, there are two major obstacles that are popping up once we try to apply these methods. The first is that actually the parameters, the variables that are measured, they are not directly related to the pathophysiology of a patient. For instance, we have usually routinely measured, for instance, a partial pressure of oxygen in the blood. But however, in the critical state like IRDS, the damage is done to the lungs, and there, for instance, the part of the lungs alveoli, they're closing, and by, for instance, the infiltrates or bacteria or other external conditions, and only after some time we see the decrease in the partial pressure of oxygen. But we don't know what's actually happening to the lungs, so we don't know the real. We can't measure the real variables that describe the pathophysiological picture quite directly, so explicitly. So only implicit markers and implicit variables. And second problem that's connected to this is actually that once we try to apply such models that are developed in one hospital or on multiple hospitals in new setting, there's usually this generalization problem that's actually quite connected to the first problem. That, for instance, if we train our models on interventions can quite significantly differ between countries because there are different guidelines, for instance, for medications. And also different populations of patients are admitted in the ICUs in the different types of hospitals, for instance. So there's different types of biases that are present in different data sets. And these two problems together, they result in the actual limitations of such models when we try to predict critical states and try to apply it later. Usually these models are quite useless. So our approach was here to apply actually mechanistic modeling, so make a hybrid model that allows us to fit the pulmonary simulator, the model, to the real patient data, and then infer the individualized parameters, and then try to utilize these parameters and see whether they're more generalizable than original variables that are measured. So that was our hypothesis. And for instance, if you see the t-SNP plot, so it's how close each of the patient trajectories are close to each other in the original data space, colored by hospitals, you see that actually the data is split by the hospital. So it means that there's a large number of biases in the hospital that tend to cluster together. So if we just look at the pipeline, so how we did it, so just shortly, so we took the different data from different hospitals in Germany, then we fitted the model that was originally developed by our collaborators in the University of Nottingham in the UK, and we also tuned it a bit so that it's this simulator can better describe the RDS state, the dynamic RDS state, and then we fitted the parameters of this model to by fitting the outputs of the model to the real data of real patients. So we fitted around more than 1,000 virtual patients, and then we compared the clustering on the original data and clustering on this model inferred parameters. For instance, among them were the number of closed compartments in the lungs, in the RDS anatomical shunt, and other parameters that are quite tightly related to the physiological picture of IRDS. And then we compared the clusterings in two cases. And interestingly, when we consider compare the two cases, on the right-hand side figure, there's two sub-figures. On the left-hand side is the clustering in the results of the clustering on original data. There were five clusters in each case. And in this case, on the left-hand side, we see that actually there's enrichment. So enrichment is an overrepresentation. So how, if you consider the cluster, how often does a patient of particular hospital belong to this cluster if compared to random sampling? And the higher is enrichment, the higher then the patients of one hospital tend to be together. And then we compare this enrichment with respect to hospital, this red bars, to the enrichment in the respective cluster with respect to clinical condition. And then we see that when we cluster on original data, there's a very high, strong overrepresentation with respect to the hospital and not to the clinical condition. So the patients of each hospital, they tend to cluster together. However, when we utilize this model derived data, then we saw that actually the patients tend to cluster. Also, there are some residual confounding with respect to hospital, but we see that there's some clusters that are only driven by clinical conditions. And then we also, it's in the paper we also investigated these clusters and found that actually they make sense from medical perspective. And that was the one of the arguments, the proofs of our hypothesis, that actually this mechanistic modeling can be utilized to infer some variables that are actually connected to the pathophysiological state, and then that can be later used in the models in the AI or for clustering or for prediction tasks or for any other tasks. So as a further work here, I can mention the work of my colleague Shadi Barakat. So here, one of the issues was that this fitting, it took quite long time. So fitting of the one patient was taken around 1 day on a single core machine. But then again, if you feed like thousands of patients simultaneously, so it took on average like several days. And therefore, there was an idea to, like, if you would like to utilize such models in the real hospital setting, then it's not acceptable these times. And the idea was to build a surrogate deep learning model to replace the simulator and check whether it can be done and how, and then the fitting of such models can be done very fast. Then my colleague implemented this deep learning model and compared the performance. And the second further work is work of my colleague Katerina Nikulina in the University of Aachen. So they're now trying to embed also other types of data, so not only dynamic continuous variables, but also imaging to check whether it adds additional information to the model, and we can then identify the parameters with more precision then. And then it finishes my presentation. Thanks to all my collaborators. And again, thanks for you as well for listening and for the invitation. And I'm happy to answer your questions. Thank you.
Thanks a lot, Constantin. Yeah, thanks. Yeah, very nice presentation. Amazing, fascinating the work, really interdisciplinary, really useful. Anyone for any questions for Constantin? Yeah. Chat. Please don't be shy.
So we don't have yet any questions in the QA box. I wonder, Mariana, if I could ask a quick question to Constantin.
Yeah, thanks a lot. Yeah.
So you were mentioning earlier that in the clustering you identify not only clusters associated with different hospitals, but also that in your cluster analysis you identify pathological conditions, the patient's conditions that actually made them cluster in different ways. Could you elaborate on that? And could you tell us whether that was actually informative from a clinical standpoint, or it was sort of relatively trivial, right? And so meaning that, yeah, you identify the cluster, it confirms your ability to simulate the conditions properly, but the information that you gather was actually whatever the clinical team could observe relatively easily via monitoring.
It's a very good question because usually, yeah, if one applies, for instance, some deep learning models for clustering for embeddings, then one usually sees some triviality like neonates, newborns, they cluster separately from adults. But in this case, I think the truth was somewhere in between. So here I didn't mention one point that because yeah, it's out of the scope of the presentation, but the thing is that we couldn't recognize real RDS patients in our data because of the implementation of the criteria. It's hard to retrospectively to really identify who's RDS and who is not. Therefore, our cohort was suspected RDS. And then, yeah, we simulated it, and then we found the cluster on this model-informed data that actually was enriched by the real RDS, by the diagnosed RDS, and that was actually very interesting because usually the problem with all this retrospective model development, and at least in RDS, it's that there is no gold standard. You couldn't implement this definition so that there's no training data for your models, for instance. And this could be also used, this modeling can be also used to try to find patients who are similar, who cluster together with diagnosed RDS. And then they, with high probability, can be also RDS patients. But then again, for this to validate, we wanted to have this step like later to validate by internists to check the data retrospectively. But that's really a good question, actually. As that, do we see some non-trivial that can be used, can expand our knowledge in the medical field, for instance, or is it just this? Is that what's expected? Yeah.
So do you anticipate that in the future, your study could evolve into predicting the time course of the pathology, essentially, predicting actually folks that are going to develop fully the pathology?
Yeah, that was our initial aim, to actually develop a system hybrid modeling for predicting IRDS, and then also like actually in the trial to prospectively validate it. But then, of course, as usual, is somehow then shrinked to some particular questions. But then, yeah, it's our maximal aim was to, of course, to utilize it in the clinic and to implement it as an early warning system and to help to augment what we can achieve with the database models.
Thank you.
I think we have some in question in QA. Do we have time to read the question from Caitlin Hall?
Yeah, why don't we do that? Yeah, I think we have the time.
Yeah. I don't know if you can see, Constantin.
Should I read it then?
Yeah.
I believe you stated that one of the contributions of this work was to infer individual parameters to approximate disease states of patients. Can you talk more about the parameters you were using for detection and how you determined their relevance to various disease states? So here thanks for your question. So here we approached it twofold. The first was to just think a priori with domain knowledge, which parameters of the model can play a role in the development of RDS. For instance, I mentioned this number of closed compartments. So it's basically what's the part of the lung is closed and it's not functional due to infiltrates that are happening during the RDS. And then we included, for instance, these parameters, and then also other parameters that potentially can play a role that we identified through the sensitivity analysis on the outputs. And by this, we defined the list of 10 or 12 parameters that we in the end fitted fitted in the optimization process. So it was like both types of from two sides.
Interesting. Yeah. Okay. If there are no other questions, maybe we can move to the next talk. Thanks a lot, Constantin. Yeah. It was really nice to meet you. Yeah. Right. The next talk is from Professor Thomas Quatieri. Yeah, and there is also his collaborator, Jing Wang, Dr. Wang. Yeah, they are from Mid Lincoln Laboratory and Harvard University, and also Richard Delora. Yeah. Thanks a lot, Thomas, and welcome.
Thank you. Thank you, Paulo. And can you hear me? Can you see my slides?
Yes, I can. We can.
Thank you. Okay, thanks, Paulo and Mariana, for this invitation. It's truly an honor to be here today. What I'm going to be doing today, along with two of my collaborators, is discuss some work that we're doing at MIT Lincoln Laboratory in the area of vocal biomarkers for screening of PTSD. Before I begin, I'll say a few words about myself. I'm a senior scientist at MIT Lincoln Lab. I'm in the Human Health and Performance Systems group at Lincoln. I also hold a faculty appointment in the Speech and Hearing, Harvard-MIT Speech and Hearing Bioscience and Technology program. And as I mentioned, I'm here today with two of my collaborators, Jing, Wang, and Delora. They are two of many authors on the paper that Mariana showed earlier. So let me begin by defining or giving you one definition of PTSD, post-traumatic stress disorder. Okay. In this definition, a mental health disorder some people develop after experiencing or witnessing a life-threatening event like combat, natural disaster, a car accident, or sexual assault. One of the implications of PTSD and the traumatic event that led up to PTSD is that this event is often relived, and this significantly impacts the quality of a person's life. Now, I think we typically associate PTSD with the military. However, we know that PTSD is prevalent in both civilian and military populations, impacting nearly 12 million adults in the US each year. Now, in the context of this particular work, we have focused on the military population, although, as I said, it's applicable to both civilian and military populations. One statistic in the military world is that 83% of all US veterans and active duty service members have experienced PTSD as a result of their service, a pretty significant statistic. So how do we identify PTSD in a human? Well, there are current assessments that rely on typically clinical interviews and self-reports used by the clinician, but there are many challenges here. First, there's a limited number of professionals who can do this kind of assessment. The screening often takes long, 30 minutes, and thus does not scale very well. They tend to be subjective, and in fact, clinicians don't always agree. And you get infrequent snapshots that don't give you a really full picture of a person's daily routine. So given these challenges, our goal is to design an objective, non-intrusive, and continuous means to detect and monitor PTSD. If we can achieve this goal, then we'd have a tool to assist the clinician in diagnosing the disorder. And I stress assist, we're not trying to replace the clinician here. And also a tool that helps us to track therapy. It could be behavioral therapy, a drug therapy, whatever. Meeting this goal could also provide us with a self-screening tool by the individual, by the participant, for early detection. So the approach that we're taking to try to meet this goal is to objectively measure changes in two characteristics of the human. One, how do we communicate, i.e., patterns of speaking, how they change, and how changes occur in how we feel, i.e., our emotional state. So these are the two properties of the human that we're trying to get at in identifying PTSD. Now, we can think of this, given that this session is on digital twins, we can think of these these properties of the human that we're trying to get at as one component of a much more complex computational model. We call this the digital twin. We want a digital twin to be bio-inspired. And we want the digital twin, this computational model, to mimic the human, a very tall order, right? All the brain-body interactions of that human. That's not possible at the moment. And for the work we're doing, we're focusing on, as I mentioned, emotion and speech. We want to understand how a person feels, the changes in how a person feels, and model that change, and changes in how a person speaks and model that change. And we derive these changes, the parameters of these models, from behavioral measurements that we make on the human. And with these parameter changes, then we can design a system that can alert the user, alert the clinician to the possibility, the risk of PTSD. That's the big picture. So let's talk about speech first. I've used the term vocal biomarker. One loose definition of vocal biomarker is that it's a measurable characteristic in the speech signal that enables insight about the physical or mental state of a person by capturing changes in sensory, motor, and cognitive function. I like to think of speech production as consisting of three different essential subsystems: respiration at the lungs, phonation at the vocal folds, articulation in the vocal tract. And we can estimate parameters that represent each of these systems from an acoustic signal that we measure with a microphone. For respiration, we can get at the envelope, which reflects loudness. For phonation, pitch, which is the rate of vibration of vocal folds. And for articulation, the resonances of the vocal tract, which shape the sounds and create different phonemes, different consonants and vowels. Why do we, why do this? Why look at vocal biomarkers? Well, the limbic system, which is one component in the brain, as you know, is hypothesized to be affected by PTSD. And furthermore, it's hypothesized to modulate the speech production system. So changes in the limbic system will change the speech production system. Let's look at one example of what I mean by a speech feature. This relates to articulation. Ideally, what we'd like to do is measure the movements of the tongue, the lips, the jaw, the velum, which are the so-called articulators in the articulation system, and use those measurements as parameters. We don't do that. We infer that, these parameters, from acoustic measurements that we make. Okay, but let's just take a look at sort of the underlying physiology here and the production of sound. What we're seeing is the lips, the jaw, the velum. With the velum is a little flap that closes and opens the nasal cavity. We're seeing changes in all of these parameters in the tongue as a function of time as a person is speaking. Unfortunately, the audio is not working here for some reason, but you can see the movement of the articulators as a function of time. It's these kinds of parameters that we're trying to get at, changes in these parameters that reflect a change in the brain due to the presence of the condition of PTSD. And what about emotion? Emotion is the other block, the other module that I mentioned within our digital twin that we're trying to get at. I think we all intuitively know that emotion comes in discrete forms: happy, sad, angry, frustrated, dysregulated, and so forth. Okay, those are discrete states. But we can map these discrete states into a continuum along two-dimensional axes. It's a continuum in a two-dimensional space, which we call arousal and valence. Arousal means excited to calm. Valence means positive to negative. Okay. What we've done is we've designed a detector that can predict these levels of arousal, levels of valence. And we could use that as parameters, if we like, in doing detection. But what we found was even more effective was that we can use these parameters to modulate or choose what segments of speech we analyze. We found that actually to be more effective than using these levels as parameters themselves in a detection system. Now we did this under the original hypothesis which was intuitive to us, that PTSD should be more apparent in regions of speech that are high with high arousal, negative valence. And in fact, we found the opposite. We found that PTSD is more apparent in regions of speech with low arousal and high valence. Okay? More calm and more positive. Those are the regions that brought out differences between the sort of, baseline, state of the human, the healthy state of the human, and the PTSD state of that human. So this is putting it all together. Here's the big pipeline. We take a speech signal with the acoustic signal. We do a quality assessment here to decide whether or not we want to enhance the signal. We measure low-level features. These are the features I mentioned earlier: loudness, pitch, resonance of the vocal tract. We then use these to predict the level of arousal and valence of each segment of speech as a function of time. We design a filter, pick out those regions that are low valence, high arousal. I'm sorry, low arousal, high valence. We then go one step further and extract features that bring us closer to the underlying neurophysiology of the human. I'm not going to go into the details here, but then these features are used in a predictive model. That's the basic idea. That's our pipeline. Now, to test this system, we need data. We've got data from 3 different sources: 2 military databases from military populations and 1 from civilian. Today, I'm going to talk about 1 military population-based data set that was collected at Walter Reed Medical Center, 144 subjects, and reading a protocol that we designed with Walter Reed speech pathologists. So quickly, how did we do? Well, I haven't talked about the details. We can talk if people have questions later. But we designed a detector, actually based on a very simple Gaussian mixture model-based detector. And what we're seeing here is the false alarm versus probability of detection curve, the ROC curve. With emotion filtering, as I described, we're getting an AUC of 0.8, which is pretty good. If you don't do any filtering, you lose performance, and that's in the yellow curve. If you do the filtering that I described according to the original hypothesis, pick off segments that have high arousal and low valence, you do about as well as doing no filtering. Another significant drop in performance. So it was interesting, and I think a very important discovery, that we could modulate speech segmentation with emotion and gain significant performance. There were very important regions in the speech signal that kind of key in on the differences between control and PTSD. How do we use this? Well, this is one concept of operations. We give a patient a tablet with our smartphone with an app upon a health check-in. The patient submits the voice recording by following apps on a prompt, prompts on an app. We obtain a detection level, a prediction PTSD level, which provides us a screening result. And this is forwarded to a clinician. The clinician then utilizes that screening information to trigger further mental health investigation. At the moment, we have this entire process on a laptop running in near real time. Takes about 4 minutes to actually give the recording and obtain a result, a predicted result. So, in summary, what have we done today? We've shown that vocal biomarkers and the emotional state can be considered aspects of a more complete digital twin of the human. Vocal biomarkers modulated by emotion was demonstrated as an effective means to detect PTSD. We are planning to use the same approach in other conditions like human depression. Clearly, we need more additional testing and validation, which is now being planned with larger and more diverse data sets. As I mentioned, we have an operational PTSD screening tool prototype that was built on a laptop and ready for clinical evaluation. I should point out that the detectors that we have developed up to now are based on group models, i.e., we're not looking at a personalized model, which would take us closer to a digital twin representation, right? So one of our future steps is to expand this approach to personalized models, if we can get more data, implement this on mobile devices, not laptops, so it's more portable, more usable. And this would bring us, the personalized models would bring us further to this, this vision that we have of a digital twin. And I want to acknowledge the U.S. Army for supporting this work. Our other Lincoln Lab team members, including Jim Williamson, Jonathan Taylor, Haley Reynolds, and our collaborators, who are all authors on the paper that I mentioned, Mariana mentioned earlier. Okay, I think that's what I have.
Thanks a lot. Thanks
Any
a lot,
questions?
Professor Quatieri. Yeah, really fascinating talk. I had a question, but you already answered to my question in one of your slides. Yeah, I will leave first to the other if they have any question. I see a hand up in the panelists group, maybe. Yeah, Constantin. Yeah, please.
Yeah, thanks a lot, Thomas. I'm really excited when I see really these applications to the healthcare. So it's really interesting project, and I would have two questions. The first is that you analyzed the voice recordings of when the person was in particular emotional state, as I understood, if I understood correctly. So if for instance a person comes to the doctor and uses this app, and he or she is in another completely different emotional state, that it's hard to extract this feature. So have you encountered that? And the second is just short, is also, are there any additional open data sources that can be used for training these models, or shall it be only limited to these data sets? Thank you.
Great questions. I'll let Jing and Rich chime in as well here, but to answer your first question, the data, well, the Walter Reed data set was collected in a very controlled environment. Right? There was an interview. It was a clinical interview in a sound room. Okay, very pristine environment in a sound room. The protocol consisted of reading a paragraph, answering some questions that provide free speech. And so, we weren't moving from one emotional setting to another, right? I think that's what you're asking. If you go from one setting to another, and we were sitting, the subjects were sitting in a sound room with a clinician, just going through this protocol. And so the emotional state of that person, it comes out in the reading of the passage. It would come out in the dialogue with the clinician, answering questions with the clinician. And we're picking off regions in that signal that correspond to a certain emotional state, low arousal, high valence. I mean, if you had trained the system in that environment, and then you go and you test it and use it, say in the home, in the community, then I think you raise a good point. You're in a different environment, and the emotional state could change. But the hope would be that by picking off those regions of the signal that have high, low arousal, high valence, you'd be consistent with the training that trainings. But that we'd have to test that. We'd have to test that. I think that's what you're asking. Yeah, and the other, what's out there? Well, there were three data sets that we have collected or worked with others in collecting, which is the Walter Reed data set. I don't think that's, I mean, we could definitely inquire about that. I don't know if it's open source. I don't think it is at the moment. It's tricky to get these open source, especially for PTSD. You have to look into the IRB. There's also the Strong Star data set, which there were three data sets. That was another military data set. That was collected at the San Antonio PTSD Center. That's a military center for military, service people, PTSD data collections. I doubt that's open source. I doubt it's open source. I think it would be wonderful if we could get these to be open source. The third one is based on a civilian first responder data set that was collected by Deborah Bridle Professor Deborah Reidel's group at University of Central Florida, I believe. That could be open source. I don't know. I can acquire on all of these. Those are the three that we have at the moment. There's another one called the Aura data set that we're about to analyze. We're getting data as we speak from the MGB McLean Center for Mental Health in the Boston area. And again, I don't know if they're making that open source. We'll see. But that's a great. Ideally, we would like to see that so other people could use the same data sets. I don't know if Jing or Rich have anything more to say or other questions.
I can. Yeah. Sorry. Yeah. Please.
Oh, sorry. If we have time, I think there's one thing I want to add for the emotion filtering aspect. Exactly what Tom was saying. In terms of if a subject is in more aroused or more negative valence state, the way we're handling right now is we're computing relative valence and arousal within the person. So when you come in a certain state, we compute relative to yourself. Are you being more aroused or calmer or more pleasant or less pleasant on that continuum? It's not absolute definitive state. So even if the protocol is moved to a different setting or the person is in a different emotional state to start, the hope is that if for a patient with PTSD, it's been clinically observed that they are less likely to experience moments of calm relaxation. So even if for that patient, we try to find the more positive valence and low arousal state, in the absolute emotional space, it probably still more towards the negative valence, the higher arousal state compared to a healthy patient. A healthy subject. So on the population level, using a population model, that the model should still be able to identify a patient's voice pattern.
Yeah.
And I'll just add one more comment about your initial point about where the data come from, how much data you need. If you look at that processing chain, every step in that processing chain has confounders associated with it. And in order to deal with those confounders, you do one of two things. You either establish a protocol that eliminates confounders, but the protocol matches how you're going to use it clinically. So you have a notion, how am I going to use this clinically? And establish a data collection protocol that matches that clinical use to eliminate confounders. Or you need an enormous data set because there are a lot of confounders. And we're struggling with this right now, I think. This is something, you can never get enough data, right? So this is a point of a lot of discussion among us and among the clinicians we work with, how to address that need for data. It was an excellent point you bring up.
And to follow up on that, one confounder being human depression. Human depression often occurs with PTSD. So you could ask yourself, well, how are we detecting the combined state of that person? You could argue, well, that maybe it doesn't matter. You're still detecting a condition of importance, comorbid conditions of importance. But that is an issue. And what is causing the change in voice when two conditions are happening at the same time? Is it depression? Is it the PTSD? Can you cluster? Can you filter out? Can you cluster features that determine which features are associated with which condition when two are happening simultaneously? We're also looking at how do we cluster features if we're detecting depression versus detecting PTSD? The features change. Do they cluster differently? And do they represent therefore different subsystems? The different subsystems that I mentioned earlier, does one become more important than another in a certain condition? Do they overlap? Are there different kinds of changes that occur within a subsystem for a condition? Those are all really intriguing questions that we're currently considering, which goes along with Rich's comment.
Thanks a lot. Yeah, I think for the timing, there are some questions in the chat. If you can, I think, yeah, some of them have been already more or less answered. If you can answer like writing, will be nice. So we can move to the next talk. I'm just saying that one for the time. Yeah. Thanks a lot, all the group, yeah, and we can move to Samon Kazemi, yeah, for the last talk. Yeah, thanks. You can start sharing your screen, yeah. Yeah.
Yes, hi everyone.
Hi, Samon, yeah.
All right.
Perfect. I can see your screen, yeah. Try to put, yeah.
Hi everyone. Thank you for the opportunity. My name is Samon and I'm a third year PhD student who works in computational medicine lab under Dr. Rose Farah's supervision, which she's also here as well. So today I'm going to present our new published work, Bayesian inference of hidden cognitive performance and arousal state in presence of music. So there is this famous law in the psychology called Yerkes-Dodson law, which notes that a moderate range of arousal can result in the maximized performance, which would allow us to enhance the performance within our everyday life routine. And then we can come up with this question that what if we could design a closed-loop architecture to let us stay in that ideal region and level of arousal within our everyday life routine. So the aim and the goal of our research is to design a closed-loop architecture to first decode these arousal and performance brain states from the available behavioral and physiological signals so that it can be implemented within the wearable devices and then implement a safe interventions such as music to regulate these states. So we focus on a working memory experiment called MBAC task, which a participant is supposed to recall the nth previous stimuli. So one back task is the easier version compared to the three back task that we basically had in our experiment. There were two types of interventions: calming background music, which is supposed to simulate the low arousing condition, versus the exciting background music, which we expected to simulate the high arousing condition. So to decode the hidden performance state, we simply use the available behavioral data, such as sequence of correct, incorrect responses, which is binary, as well as the reaction time, which was continuous as the available observation. And then we pass that to our Bayesian type of decoder to estimate the hidden trajectory of performance state in a continuous manner so that we can have the dynamic of the performance as well. So another state of interest is the arousal state, which also previous presenter also mentioned. So the arousal state basically is highly associated with the pseudomotor activity, and then a skin conductance signal is also one of the informative biomarkers that can be used to decode the hidden arousal state. So the way that we handle it, we deconvolve the skin conductance signal to recover the underlying basically brain activity. And then once we recover these underlying brain impulses, we use them as the observation to decode the hidden arousal state in the continuous manner. So now that we have the arousal and performance data points, we wanted to see what is the actual link between the arousal and performance. And then we also wanted to investigate the feasibility of inverted EULA within the data points. So we use this quadratic function to model the inverted EULA. And then what we have seen was a decent alignment between the observed data points and then the fitted basically model, which would inspire us basically to use the performance coupled with the skin conductance signal as the observation to decode the underlying arousal. So ideally, this decoder can be more emotionally intelligent in the sense that it accounts for the concurrent performance as well. So here I'm showing our results, and then the green background color is indicating the calming session versus the red background color is showing the exciting session. The first sub panel is showing the collected skin conductance signal. And as you can see, the baseline of the skin conductance start to decrease during the calming session. The second sub panel is the recovered brain activation, which during the exciting session, the number of impulses as well as their amplitudes are slightly higher. Then the third and fourth subpanels are the behavioral data that we have used to decode the performance state, which is shown in the fifth subplot. And then the last subpanel is showing the decoded performance-based arousal state. And what is actually interesting is that, so we can see that in the region that the arousal is low, the performance is also low, which may be an indicator of conditions such as boredom, while the region that the arousal maintains a moderate level, the performance is high. And on the other hand, when the arousal is too high, which again can be an indicator of a stressful condition, the performance is low. On the right side also, as expected, this new decoded arousal state can depict a more aligned arousal performance linked with the inverted EULA, which can provide an opportunity for basically implementing refined control strategy. And to summarize, we developed decoders that can be used to track these arousal and performance hidden brain states. And then we investigated the feasibility of music as a stimuli to regulate arousal and performance. Then we evaluate the possibility of performance by setting the arousal in a moderate range. And then in future, this research can be used in educational setting and smart workplaces to increase the productivity and performance. So with that, I would like to thank you for listening to this presentation and let me know if you have any questions.
Thanks a lot. Sorry, thanks a lot, Samon. Yeah, really fascinating as well. Brilliant talk, really inspiring research, very useful in the psychology side. Yeah. Any question? I know that we're a little bit late, but I think we got 5 minutes for any questions for this group and this study. Someone has
Oh, I was wondering, did you find is there any particular kind of music that is more useful than another?
Oh, the type of music that has been used. So that's a good question, actually. So we asked the participant to provide the music that it sounds calming or exciting to them, so that it would be personalized and person specific. So they provided the music based on their taste, even though it can also produce a basically familiarity impact, which can be one of the confounding factors, but in a sense that it would be more personalized, we use that to be in a real world scenario. So one way to address that familiarity in future, we did that experiment actually. So we get the music from the participant and we use the basically deep learning model to generate the new music.
Oh,
yeah.
But
Yeah.
not.
Sounds a great idea. Great idea to use music in that way. Yeah.
Mm-hmm. Yeah.
There was one question in chat.
I think he's referring to the previous one. I don't know. In the question and answer, let me see the chat. The chat, no. I think the Esteban asked for the previous talk. Yeah.
No, it's the one that I see. It says, "Can you explain more about the emotion filtering that you mentioned?"
Yes, yeah, that was referred to the previous one, but of course this maybe can apply to you as well. Yeah.
Yeah, sure.
Okay.
Let me find my slide again. So the filter that we have used to decode the arousal was basically we deconvolve the skin conductance signal first to recover these basically impulses, and then we use these impulses. So these impulses have the onset and as well as the amplitude. So we consider both of them as the informative observation. And then if we form a marked point process type observation, and then we pass that through the Bayesian state space decoder so that there's no need for the training session, and then we decode the arousal.
Yeah. Any other last questions for Samon? Yeah.
All right, then. Thank you.
Yeah, perfect. Thanks a lot. Yeah. If you have any question, just contact me and I can put you in contact with the authors if this is okay. Yeah, for them. Yeah, thanks everyone. Yeah, I will put my camera on at this point, maybe. Yeah, okay. I don't know if it works. Looks doesn't work. But yeah, sorry for that one. Okay, so thanks again for joining me and joining Paolo for that brilliant, I think, webinar. I hope really enjoy it, and you can read all the papers, and soon will be also an editorial about these 7 papers in the special issue. Yeah.
Thank you very much. Thank you, Mariana, for putting together this excellent special series that we publish in IEEE OJEMB.
Yeah.
Thank you for running the webinar today. And I look forward to seeing most of the attendees, I hope, at the EMBC next week. Thank you very much.